Interning at the Microbial Ecology of Terrestrial and Aquatic Systems Laboratory, or METAS Lab for short, at the Institute of Biology, UP Diliman, was a milestone in our college journey as BS Biology students from UP Manila and Los Baños. What began as a summer internship quickly evolved into a transformative learning experience, an opportunity to grow not just in skill, but in mindset, collaboration, and curiosity. Within the first few days, we were immersed in the daily rhythms of a working microbiology laboratory. We practiced foundational techniques such as streaking, plating, pouring media, isolating microbes, and optimizing growth conditions. While these procedures may seem routine, they taught us discipline, consistency, and the importance of mastering the basics before moving on to the complex. Each action, every pour, every streak, was a lesson in precision. We learned that science demands not only intelligence, but also patience, attention to detail, and respect for the process.
As the internship progressed, we moved into a more advanced molecular biology techniques. We performed polymerase chain reaction, or PCR, a method used to amplify DNA, allowing us to analyze microbial genetic material. This was followed by agarose gel electrophoresis, where we visualize DNA fragments, gaining insight into gene presence, size, and amplification quality. We even delved into virulence testing, helping us understand how microbial strains might contribute to disease, a critical concept in both environmental and medical microbiology. But what truly pushed our boundaries was being introduced to molecular docking. Here, we studied how small molecules such as toxins or other therapeutic compounds can interact with microbial proteins at the atomic level. We explored software that allowed us to model these interactions and proposed novel research topics using molecular docking for applications in environmental diagnostics, public health, and marine rehabilitation.
Outside the lab, our learning continued. We attended graduate seminars where master students presented their proposed thesis topics. It was fascinating to see how diverse biology could be, ranging from conservation genomics to host pathogen interactions to the ecology of mangrove systems. These seminars were more than just lectures. They were windows into the future of scientific research. We observed how projects are designed, how questions are formed, and how ideas evolve through critique and discussion. We also held journal reporting sessions where we critically analyzed published papers and reflected on their methodologies, results, and implications. This helped us not only understand how to read scientific literature, but also to identify gaps, ask better questions, and communicate findings with clarity and confidence.
A standout experience during our time at METAS was our introduction to cetacean pathology in partnership with the IESM. We explored real-world applications of biology through necropsy observations, marine mammal tissue analysis, and discussions on blast trauma, acoustic disturbances, and their effects on whale and dolphin populations. These experiences emphasize the role of cetaceans as sentinel species, organisms that reflect the state of the environment. Understanding their biology allowed us to appreciate the interconnectedness of marine ecosystems, conservation science, and human impact. These moments reminded us that science is not just about understanding life, it's about protecting it. We also learned about the importance of lab management, from maintaining inventories and ensuring proper storage to preparing reagents and managing lab wastes. These behind-the-scene tasks were vital in ensuring the laboratory functioned smoothly and responsibly. This exposure reminded us that being a biologist isn't just about conducting experiments. It's about upholding standards, ensuring reproducibility, and working as part of a team committed to the integrity of science.
One of the most empowering parts of the internship was being encouraged to propose our ideas. Some of us explored how docking could be applied to design antivirulence agents, quorum sensing among fermented foods, while others envisioned studying protein structures from harmful algal blooms. The lab didn't just give us answers, it gave us the tools and confidence to ask better questions. Through every day in the lab, every discussion, and every experiment, we were not just learning techniques. We were learning how to think like scientists. We made mistakes, we asked questions, we celebrated breakthroughs, no matter how small. In just a few weeks, the METAS lab taught us what textbooks alone never could: that science is dynamic, collaborative, and rooted in purpose. It gave us a glimpse into what lies ahead and the responsibility that comes with knowledge. This internship didn't just train us in techniques, it shaped our direction. It helped us realize that wherever biology takes us, be it research, conservation, public health, or education, we are ready to take that path. Because at METAS, we weren't just interns, we were scientists in the making.